Salam kebajikan. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Distinguished guest lecturer, Professor Roxana Philip, and all dear students. My name is Indah Novita Sari Sudarsana, and I will be the moderator for today's session. So it is a great pleasure of mine to introduce our guest speaker for today, Professor Roxana Philip. So Professor Dr. Roxana Philip, MD, PhD, is a professor at the Faculty of Medicine and Biological Science at Stefan Cel Mare University of Suceava, Romania, Europe. Also act as the head of the Tuberculosis and Molecular Biology Laboratory at Suceava Emergency County Hospital in Romania, Europe. Professor Roxana graduated from the GRT Popa University of Medicine in Romania, finished her specialist in clinical laboratory medicine in 1995, and earned her PhD in microbiology under the supervision of Professor Dumitru. For over 30 years of experience, Dr. Philip has held many academic research and position in Romania. She has been an invited speaker and facilitator at major ESC-MID events and medical institutions all over Europe, Latin America, and Africa. Also played a key role on Romania's early COVID-19 response, and also actively involved in national and European research projects. With her tremendous experience, we're going to learn about antibiotic resistant mechanism, which is a very important topic in this modern medicine. So please allow me to welcome Professor Roxana Philip. Professor, the time and screens are yours.
Good morning to everyone. I'm very happy to be again with you, the dear students and staff from Ganesha Medical University, where last year by this time I was guest, and also I discovered the beauty of your island. And here in my office I have my picture from the Tanakh Lot temple that I remember with great pleasure. In our hospital, as you can see, I'm dressed in the laboratory coat because in our time is 9:30 a.m. when we have started our activity, while in your country is already noon time. The topic of today is a very hot spot in the microbiological arena because it speaks about the bacterial resistance against antibiotics. And according to my experience, I got discussions and lectures in 3 continents, Europe, Africa, and Asia. And wherever I was, the antimicrobial resistance problem was the same, meaning that the bacteria produce a lot of new resistance mechanisms, while the antibiotic resources are very limited. And I'm sure that you are aware of this, and I have visited the laboratories in Surakarta, and I have seen that you have very nice methods to identify the resistance mechanisms. As your moderator informed me, you are students in the first year of study from the medical faculty. Is it right? Am I right? Because I want to see your background, the microbiological background. What is the background of of this audience? Please feel free to answer. Someone from the audience, please. Is it possible?
Jadi mungkin ada yang mau menjawab sekarang semester berapa gitu. Profnya ingin
We're
tahu.
on a second semester, Professor.
Second semester in the first year of study, yes?
Yeah.
Have you studied microbiology?
A few lecture about microbiology regarding the basic of microbiology and
Not yet.
maybe
So we
a little
can,
about yeah.
Okay, so this will be a very good trigger for you to study more on this subject. So, Miss Moderator, I will share the presentation now. I will do it on the screen as everybody can see it. Can you see it, the presentation? Can you
Yeah, we
see the
can see it,
screen?
Professor.
Inda? Yes. Okay.
Yes.
I will talk about the antibacterial resistance mechanisms. I will give you an overview, and I'm sure that this will open your curiosity because I'm not going to cover everything because it's a very huge topic. But on the other hand, you have the possibility to establish new ideas, and I'm open to receive and to be in contact with you for your ideas. So I'm currently employed in the Faculty of Medicine and Biological Sciences from Suceava, which is a city in the northern part of Romania. And also I'm appointed as head of the department in the Molecular Biology and Tuberculosis Diagnosis Laboratory in the Suceava County Emergency Clinical County Hospital, which is a 1,400 facility covering all the specialties, also thoracic surgery, plastic surgery, pediatric surgery, neurosurgery. So these specialties, which are only mainly in big hospitals. Why do I say to you this? Because you must understand that the etiology of infection spectrum is very large. So we cover a lot of specialties, but also a lot of pathology, starting with, let's say, Staphylococcus aureus and ending with other agents, like, for example, Legionella, which we identify by molecular methods. What are the main things that I want you to remember? This meaning is antibiotics are tired, but not the bacteria. Meaning what I've told you before, that the antibiotics can't be discovered in the same rhythm as the bacteria produce resistance mechanisms. In the last years, it has been discovered only one antibiotic class, but if you look in the literature, the bacteria have been discovered a lot of resistance mechanism, and not only one resistance mechanism. In a bacterial strain, we can have the interplay of resistance determinants. That means associations of two different resistance mechanisms. For example, enzyme production and porine modification. What is the practical outcome of this thing? Anak, agung, bagus. Do you know what is the practical input for this?
Yes, Professor.
Coexistence of two resistance mechanisms in a bacterial strain. Which is the practical impact?
From what I learned, maybe a little about how they gain resistance. So basically, they have those chromosome that on the bacterial and the outer chromosome that is known as plasmid. So the plasmid can exchange information between the actual chromosome of the bacteria and actually the extra chromosome of the bacteria itself, known as plasmid, and can be transferred from one another. So that's how they gain resistance through the process of conjugation, transposon, and maybe transformation if there's a leakage on the chromosome, like bacterial getting lysis, destruction. So the other bacterial that's not having those, ability to help with that. So they actually just take on that resistance to themselves by applying what the others have. Like that.
Yes, you impressed me. It's a very good answer. But what I want to underline is chromosome is present in all the strains of a bacterial species, while the plasmid is mobile genetic element that can be transferred either between gram negatives or even though between gram negative and gram-positive. Yes, you are right. You described the molecular mechanism, but the question was, which is the practical impact? The patient is not interested to see which is the bacterial species. He is interested to have a treatment for recovery. So if bacteria have several resistance mechanisms, this complicates very much the treatment because we have to associate different antibiotics, and that means more expensive and also the possibility for gaining resistance under treatment, that is inducible mechanisms. And there are certain antibiotics which can induce resistance under treatment. So I treat the patient and the strain become resistant. So the practical impact of the association between resistance mechanisms is that this complicates the therapeutic strategy. So in this situation, there is a question: what's left in the cupboard? This is a more poetical approach, but what does it mean? Which are the antibiotics that we can use for treatment, okay? And there are 3 points that I want you to remember, and you can make some notes. Bacterial resistance from one side, discovery of new antibiotics, the other side, and antibiotic stewardship. What does it mean, antibiotic stewardship? Means antibiotic treatment is administered after certain criteria, meaning that we can't go in the market or in the pharmacy to buy antibiotics as we like. Do you have this possibility in Bali? Because I don't remember. Can you buy freely antibiotics from the pharmacy?
Please let me answer the question, Professor. So currently, if we want to buy a certain medication, especially antibiotic, the apotheker is going to need prescription for that. So it's not
Okay, this is a very big step forward, meaning that if you can't buy freely antibiotics, that means the resistance situation in your country is under control. I hope so. Who is interested and who is involved in this triangle? Firstly is laboratory, who detects the resistance, epidemiology, who reports the resistance pattern, and also the clinic who must give the treatment. Okay, so please remember this first triangle and also the ones which are involved. This is a print from a Journal of Antimicrobial Chemotherapy from 2011. And in this number, you see the big title, Discovery Research, the scientific challenge of finding new antibiotics. So even 15 years before, in 2011, this was the problem, the discovery of new antibiotics, meaning that from 2011 to now, there is only one antibiotic class that have been discovered. What does it mean, antimicrobial resistance? It produced almost 5 million deaths worldwide, according to the statistics of RESA reported in 2024. That's why the WHO, do you know what is WHO? This is the World Health Organization, which, as you know, is divided in 6 regions, and you belong to the Asian region, which has the headquarters in Manila, Philippines. And the WHO introduced this aware antibiotic classification. And this aware means access. This is the A, meaning that antibiotics should be readily available to treat a wide range of clinical infections. Then WA means watch antibiotics which are more likely to select for resistance, and their usage should be discouraged unless clearly indicated, meaning that when a patient got an infection, you don't have to treat with the strongest antibiotic. You have to treat according to the resistance pattern of the hospital and also according to the antibiogram results. But until you have the results from sensitivity testing, which takes you 72 hours, you can have an orientation according to the pattern in the ward or in the hospital. And also reserve, this is RE, which means antibiotics used to treat multi-drug resistant bacteria. So the conclusion is that on the first step, we have to use excess antibiotic. And also for excess antibiotics, we can't buy them freely from the pharmacy. We need prescription. This aware book has been published by WHO in December 2022 and is a very useful tool, especially for the clinicians who deal with patients and who are our partners in the diagnosis process. This book provides advice on the optimal use of antibiotics, also provides recommendations for the first and second choice antibiotic regimens for around 35 clinical infections, also including guidance on the dose and duration. And there is a strong focus on primary health care, which accounts for more than 90% of global antibiotic use. Primary health care means family practitioners, means GP, general practitioners, who are very keen to prescribe antibiotics, because if the patient comes for seeing the doctor, he needs a very quick recovery, and the GP immediately prescribes antibiotic. And this is the main point. We have to decrease this prescription as to decrease the resistance ratio. Antimicrobial resistance as a concept has multiple definitions: mechanistic, laboratory, and clinical, and represents the interaction or interplay of drug exposure, that means dose and schedule of the antibiotic. Do you know why do we have to give the antibiotic at certain hour during a day and at certain time interval? Do you know this? Can you answer? Who wants to answer? Why do we have to give antibiotics in a certain hour every day during antibiotic treatment? Normally it's 6 hours interval. I will answer you because this interferes with bacterial growth, and we have to chase bacteria exactly at the same time, because otherwise the mutants can appear. Also, a measure of potency of an antibiotic for its microbiological target and the magnitude of drug exposure relative to minimal inhibitory concentration that is required to secure a favorable therapeutic response for any given disease. This means pharmacodynamic target, meaning that we have to check if the antibiotic has achieved the minimum inhibitory concentration, which is a concept meaning the minimal antibiotic concentration that inhibit bacterial growth. Also, there is the triple lock, which is a name that shows the interdependent nature of this concept. What does it mean? In this graphic, you have the rate at which microbes are acquiring resistance, and clearly is shown that this is greater than the rate at which antimicrobials are being discovered. So in red, you have the antibiotic resistance, and you can see the sulfonamides immediately increase the resistance after the year of discovery, discovered in 35, and after 15 years, look, the resistance increase. The best one is vancomycin, which start the resistance in 1990, and nowadays represents a big problem because there are enterococcus which are vancomycin resistant. Also penicillin after discovery before 45, and as you can see, immediately resistance appeared. Probably I need antibiotic, no? What we can see in this slide is that more than 25,000 deaths are from multi-drug resistant bacteria, according to reports in 2019. That means increased mortality rates, estimated annual cost for healthcare system, and money loss, almost $300 billion. N-ai ceva decât... And also, you know, surgery, transplants, and chemotherapy are no longer viable due to infection. Which are the main antibiotic classes that we have in the present? First are beta-lactam antibiotics that act on the cell wall, bacterial cell wall mechanism. These are beta-lactams, which are the biggest class of antibiotics, and our resistance mechanism is due to beta-lactamases. Also, the second class is DNA and RNA synthesis, which is represented by fluoroquinolones and rifampicin. Then folate synthesis and trimethoprim and sulfonamides. This slide you can put together with the previous one, and you can see this correlation, okay? So when we finish this meeting, you can put this slide near this slide to see the resistance mechanism of this antibiotic. Also the cell membrane, which is daptomycin, the protein synthesis. You know where is realized the protein synthesis in bacteria? On ribosomes. And on the right side, you have the resistance mechanism. The main resistance mechanism is represented by inactivating enzymes, which is specific for beta-lactams, aminoglycosides, macrolides, and rifamycins. Rifamycins is the class of antibiotics that act on Mycobacterium, which is the etiological agent for tuberculosis. Și asta. Merci. Also, the target modification, which is for fluoroquinolones, rifamycins, vancomycin, penicillins, macrolides, and aminoglycosides. Also, on the bypass, which is represented by tetracyclines, trimethoprim, sulfonamides, and vancomycins. By the way, do you know an example of beta-lactam antibiotic? Who knows? Do you know?
Penicillin, Professor. Penicillin, amoxicillin, cephalosporin.
Perfect. I see that you have the chromosome for being microbiologist. Anak, you have the genetic material to be microbiologist. That's right. Beta-lactams are represented by penicillin and cephalosporins. Cephalosporins, which are first, second, and third generation cephalosporins, ceftriaxone, cefotaxime, and all the others, okay? Also, the antibiotics that act via efflux by porine change, like fluoroquinolones, aminoglycosides, tetracyclines, some beta-lactams, and macrolides. And now I'll give you the answer. type of resistance, the natural resistance, which is genetically determined in bacterial chromosome. That means that there are certain bacteria that have resistance anyway. So we can't give that antibiotic because per primum ab initio, this resistance is chromosomally codified. So is no chance that we can have a successful treatment. This is the case for Enterobacterales and penicillin. You never give penicillin in a patient with E. coli infection. And the natural resistance is a phenomena accelerated by the misuse of antibiotic drugs, which is reported by WHO fact sheet even in 2014. The naturally occurring resistance genes are listed here. Also, there are random mutation of biosynthesis genes providing a selective advantage to cells. Also, the co-selection of antibiotics and antibiotic resistance genes. What does it mean? Acquired resistance means that initially susceptible bacteria become resistant. And here there are two explanations. First explanation is that we are doing the sensitivity testing in vitro, so outside the human body. In vitro, in the Petri plate and in a laboratory tube is one thing, but in the human body is different. So it's very possible that in vitro the bacteria is sensitive, while in vivo it can be resistant. So this is the first explanation. And the second one that I already told you is that the bacteria can gain resistance under treatment. There are certain antibiotics which are inducers. How does resistance evolve? The answer is here. The more we use them, the more we lose them. What does it mean? We use antibiotics and the resistance appears. So this is a very good point that in your country you have the possibility to buy antibiotics only under prescription. The resistance evolved first by genetic variation, then antibiotics select for resistant already present organism. And here there is the exchange between resistance genes between the normal microbiota and the etiological agent. This leads to increase in antibiotic use and increase in resistance and colonization. Okay, here you have the diagram of the total antibiotic use, and this is the situation for strep pneumonia. And the antibiotic use presented in daily doses. And you can see the trend. Here you have Europe. You see that here you have only Taiwan from Asia, because probably they have the reports from over there. Here there are the prices, and you can see this balance. While in 2010 the use of antibiotics was 63 tons, in 2030 it exceeds 100,000 tons. So on one side we want to stop the antibiotic usage, and on the other side you can see the antibiotic usage is estimated to be very, very high, meaning increased by 2/3. And how this thing can be possible? It can be possible because, firstly, the antibiotics, 30% are consumed by humans, but don't forget the animals. 70% are consumed by animals. In the animal husbandry, to prevent disease, the animals are injected by antibiotics. We buy that meat, the product from animals, and in this way we induce the resistance. Which are the drivers of antimicrobial resistance? That means the leading forces. Which are the things that enables the resistance? First of all, antibiotic usage, biocides. That's why in hospitals we have to change the biocides at least at 6 months. Also the industry, and also the genetic. Here you have the graphical abstract of what I've told you, and is very important. Where do these products go? In the water, wastewater, discharge, aquaculture, and groundwater. So in the wastewater, you see bathing water, shellfish, we have the possibility to detect bacterial resistance. And there are studies, there are things in which we can discover these resistant strains. How do the transfer occurs? What your colleague has already mentioned is a vertical transfer, meaning that from the parental generation by antimicrobial resistance, from one individual occurs 3 individuals. 2 are okay, but 1 is resistant, okay? Horizontal transfer by the mobile genetic elements. The mobile genetic elements are more dangerous. As you can see here, I have 1 which is like the parental population, and 2 of them differently colored, which are resistant, okay? And I think here is the time that you can write something, because probably you will hear in your future activity some acronyms like MRSA, meaning methicillin-resistant Staphylococcus aureus. Have you heard about this or no? Then PRSP, penicillin-resistant Streptococcus pneumoniae, VISA, vancomycin intermediate Staph aureus, VRSA, vancomycin-resistant Staph aureus, VRE, vancomycin-resistant Enterococcus faecium, ESBL, meaning extended spectrum beta-lactamases, and carbapenemase-producing Enterobacterales, CRE. And this is a big point, at least in Europe and America. Carbapenemase-producing strains are a big concern because the carbapenemases codifies for carbapenem resistant. Carbapenems are very strong antibiotics that are kept for very serious infections. So in this situation, we have the possibility to detect the carbapenemase producing in the normal flora of the patient by doing the rectal swab. So if we are facing this situation, especially in the intensive care unit, the patient comes in a very severe condition, but normally he has been in different hospitals and he carries the resistance genes from those hospitals. So at the first presentation in our intensive care unit, we can do the rectal swab to check if the patient has already resistance enzymes, because if he has resistance enzyme, the therapeutical resources are limited, so we don't use the antibiotics with no point. Other acronyms are multi-drug resistance, meaning MDR, extensively drug resistance, and pan-drug resistant. And unfortunately, we are facing in the last years this situation, the pan-drug resistant, meaning resistant to all tested antibiotics. And in this situation, the therapeutical resources are 0. Also, there is the group, the escape group, containing E. coli, Staph aureus, Kleb pneumonia, S. netobacter, Pseudomonas aeruginosa, and Enterococcus fecalis and faecium. And you have here, first, the microscopical examination, gram-negative bacilli, and as you can see, the dangerous etiological agents are also gram-positive and gram-negatives. Gram-negative, gram-positive, gram-negative, gram-negative, gram-positive. And here you have the infection spectrum. What kind of infections do they produce? Also, the space organism, meaning Serratia, Providentia, and indole positive, also Citrobacter and Enterobacter species. All of them are gram-negative bacilli. and the other group escape, meaning Enterococcus faecium, Staph aureus, Clostridioides, Acinetobacter, Pseudomonas, and Enterobacterales. So please remember these acronyms, put it in your notebook, and when you will have the microbiology lecture and also the infectious diseases lectures, you will remember this. So here you have the overview on beta-lactam antibiotics. They started with this beta-lactam nucleus, which has been upgraded from the first beta-lactam discovered, penicillin, to the others beta-lactams, ending with cephalosporins and carbapenems. So penicillins, and here you have the classification of penicillins and anti-staphylococcal. What does it mean, anti-staphylococcal penicillins? Means that they cover also the strains that produce beta-lactamase or penicillinase, okay? Also, the anti-pseudomonal penicillins, which are represented by the association between beta-lactam and enzyme inhibitor. Also, aminopenicillins and cephalosporins. You see here there are is the classification of cephalosporins according to their generation. Do you know which is the criteria that lead to cephalosporin classification in generations? Do you know? Why the cephalosporins are classified in generation? First, second, third, fourth, and fifth generation. I will answer. Do you want to answer, Anak? Why?
Okay, I want to answer. The first and the second generation is for the gram-positive bacteria. The third and the fourth is mostly for the gram-negative bacteria. Actually, they both work on the gram-positive and negative, but lean to one side. Like for the first and second generation, lean to the positive. The third and fourth is to the negative. While the fifth generation is for the MRSA kind of type, like the multi-resistant bacteria, from what I know.
Okay, congratulations. You have serendipity because the answer is just here on the slide. And congratulations because you have the chance to see that the answer is here. So summarizing, cephalosporin classification is according to the spectrum of activity. First generation, cefalexin, cefazolin, which has a narrow spectrum, then cefuroxim, ceftriaxone, ceftazidime, cefepime, and ceftarolin, which is restrictively used. Also, beta-lactams are monobactams and carbapenems, imipenem, meropenem, doripenem, ertapenem, okay? And here in this diagram, you have the target. These are the beta-lactamase inhibitors, clavulanate, sulbactam, and tazobactam. And this is very interesting because these compounds don't have activity by their own. They have activity only when associated with the beta-lactam, like ampicillin clavulanate or amoxicillin clavulanate, ampicillin sulbactam, or piperacillin tazobactam. So only in association. You remember this. So from this slide, you have to remember the cephalosporin classification and the resistance mechanism. Here you have the target of the beta-lactamase. As you can see the name of the enzyme, they destroy the beta-lactam nucleus. Which are the resistance mechanism to beta-lactam antibiotics. Here you have the structure of the gram-positive cell wall, which, as you know, is very different from the gram-negative. The gram-negative cell wall, even if it is thinner, but is more complicated than the gram-positive. Here you have the mechanism of PBP, penicillin binding protein, which act by target modification. Then here you have the beta-lactamase, like enzyme modification by antibiotic degradation. And here you have the beta-lactamase classification, and here is one of the most complete beta-lactamase classification according to Bush, Jacobi, Medeiros, because it is a functional and molecular classification. Class A and class D class A represented by SHV, TEM, CTXM, KPC, and these are acronyms, sulfhydryl variable, temoniera. This TEM comes from the first name of the first Greek patient from whom in 1964 was isolated the first beta-lactamase, and this TEM comes from his name, temoniera. CTXM means cefotaximase, KPC means Cleb pneumonia carbapenemase. Class D represented by oxacillinase. Class B, imipemenase, Verona imipemenase, New Delhi metallobeta-lactamase. And Class C, MC, cefamicin, cefoxitin, and other MC beta-lactamases. These are metallobeta-lactamases. And from class A and class D, via mutation, appeared the extended spectrum beta-lactamases. And unfortunately, in the last period, the strains had the basic beta-lactamase and also the extended spectrum beta-lactamase. So this association is killed because we don't have the therapeutical resource. And I suggest you to remember this graphical abstract because here you have the essence. And also here you have the action mechanisms for PBP from the surface protein. Everything is integrated there. And here I show you some slides, some pictures according to the sensitivity testing. Here you have the disk diffusion method. I'm not sure if you have seen this method. Is the currently used testing method in laboratory. We inoculate an agar plate, a special agar, Müller-Hinton, with the bacterial suspension of a certain density, and then we place different antibiotic microdiscs of certain concentration, as you can see here. And here, for example, you have the clavulanic acid synergy test. You have here the amoxicillin clavulanic acid, and this is placed near ceftazidime, cefotaxime, ceftriaxone, and S-trionem. And you have this special shape of the interaction between the two antibiotics. And for the laboratory, this is very important because from this picture you can infer the resistance mechanism. Another detection method is the E-test. This test combines the diffusion method and also the determination of minimal inhibitory concentration, that MIC that I have showed you before. And here you have different antibiotic concentration, and here you read the MIC, okay? And this is supplied by different manufacturers for different antibiotics or different association. Another possibility for identification is the chromid medium, especially when you are doing the screening for beta-lactamase or extended spectrum beta-lactamase production, because here you see colonies of different colors. This means different species and different enzymes, okay? And here you have the MC cephalosporinase producing Enterobacterales, and you have here the main genus, bacterial genus in which they are encountered. They are inducible, they resist to clavulanic acid and cefoxitin. So the therapeutical options is for fourth generation cephalosporin and carbapenems. Here you have the derepressed MC, ceftazidime, cefoxitin, cefotaxime, and here you have inducible MC. You see the disc of cefoxitin is placed near this third generation cephalosporin, and you have this shape of the interaction zone, okay? Also class C, the laboratory test, the Hodge test. I don't want to speak more about this. And also here you have the differentiation of different extended spectrum beta-lactamases using specific inhibitors. For example, here you have the extended spectrum beta-lactamase. Here you have the association between ESBL and MC. And here, what do you have here? This is a susceptible strain, because how do we detect the susceptible strain? We load the plate with bacterial suspension and we leave at 37 for incubation for 24 hours. And as you can see here, you have the inhibition zone. We measure this inhibition zone and we compare with a table which classified the bacterial strain in susceptible, intermediate, or resistant. I'm not sure on which standard do you work. Probably you are affiliated to CLSI. So there are different interpretation standards. The carbapenem-resistant Enterobacterales, for example, Klebsiella pneumoniae carbapenemase, and what is very worrying is the increased incidence in pediatric infections. Which are the risk factors in children? Medical comorbidities, long hospitalization, immunosuppression, prior antibiotic use, exposure to carbapenems and aminoglycosides. Which are the therapeutic options? Colistin and other polymyxin, and also oral phosphomycin. These are options for Europe. Probably you have this in your country. So, uncontrolled antibiotherapy determines resistance increase both in pathogen and in normal flora. This fact impacts hospital environment, but also general environment and everyone who is involved in patient care, medical personnel, other patients and families, and also I would say medical students, because you are visiting the hospitals. After the third year of study, you will visit the clinical ward. So you come with your microbiota, and also you can exchange microbiota with the patient. That's why you have to protect yourself and to wash your hands. Resistance spread can be realized by plasmids, transposons, and integrons. These are mobile genetic elements. The first beta-lactamase described has spread in more than 70% of Enterobacterales and 30% in Neisseria gonorrhoeae, which is one of the most important agents in sexually transmitted diseases. The majority of beta-lactamases encoded by mobile genetic elements originates in the bacterial chromosome of other species. For example, SHV-derived beta-lactamases are from Kleb pneumoniae chromosome. Also, plasmid MC expressed in Kleb pneumoniae and E. coli are almost identical with chromosomal MC of the complex of Enterobacter cloacae. The most frequently encountered extended spectrum beta-lactamases, CTXM, originates in Cluivera, according to the report of Patricia Bradford. The AMP-C beta-lactamase belongs to class C amber classification and determines resistance to penicillin. So you must understand that each type of enzyme codifies a certain type of resistance. So the significance to the clinical doctor is that if we report a certain enzyme, he must be aware that the bacteria is resistant to certain antibiotic. That's why you must understand the role of the laboratory in patient care. The laboratory is not only a producer of results that are not understood by the clinicians. Even you are medical doctors, you can be laboratory personnel or clinical personnel. In every side of this game, you must understand that laboratory plays a very important role. And you must go and visit laboratory and ask, what does it mean this? And they must explain you, because in this way you put the patient in the center of your medical care, and the patient is happy and the hospital is happy because the money are saved. You don't give treatment that is for no use. Also, another problem is Acinetobacter, which codes a lot of genes and is also resistant to extended spectrum cephalosporins. Also the Burkholderia, which have a class A chromosomal beta-lactamase and is mostly encountered in patients with cystic fibrosis. Pseudomonas aeruginosa has an MC enzyme chromosomally codified, but also is a host for different mutations, which give hyperproduction of MC resistance. Also, porine permeability changes associated mechanism and KFPM resistance. The plasmid codified extended spectrum beta-lactamases are a big concern since 1989. The TEM and SHV variants, first Klebsiella ozene, then in April, since its discovery in 1985 until 2020, were described 183 variants of TEM and 178 variants of SHV. You can visit this site if you want more information. The CTXM was first reported in 1980 and spread since 2000. Hydrolyzes cefotaxime and ceftriaxone more than ceftazidime. And the big problem, CTXM15, which hydrolyzes ceftazidime. So ceftazidime is not active anymore and is encountered in Kleb pneumonia and E. coli, which are two of the most frequent encountered etiological agents. Carbapenemases, the SME, which is hosted by Saresha Marsesens, and there are reports from England, United States, but also in Europe. Also, OXA 48, 23, and 40, which hydrolyzes penicillin and carbapenems. So you can see we give carbapenems and they can still are inactive. That's why it's so important that the laboratory detects and report these problem strains. And nowadays we have possibilities for molecular diagnosis, and we can report in less than 2 hours the strain and also the resistance mechanism. Here I select you some antibiograms, but I'm not sure that you can see it. You are supposed to see here the inhibition zone. Here, I think you have a better view for an E. coli, which is susceptible to all the antimicrobial testing. Here, this is penicillinase producing strain, which is resistant to amoxicillin and ticarcillin, as you can see here. Yes, you have sensitivity here. The diameter is bigger. Also, the cephalosporinase phenotype, which is resistant to amoxicillin, cephalotin, and association with clavulanic acid. Okay. Here we come. You remember the 3 points that I showed you before. One of it is antibiotic stewardship. What does it mean? This is an important part of another concept of diagnostic stewardship. What are the recommendations of the Infectious Disease Society of North America? Recommends audit on antibiotic consumption and a team that immediately provides feedback to the clinician. Do you remember I presented you the team, laboratory, epidemiologist, clinician, okay? And latest is also the pharmacy, the clinical pharmacist, who has also a very important role. Also, restricted use of existing antimicrobial agents by revision or request or special authorization with monitoring the evolution trends. And here is the most important role of the epidemiologist. Which are the supplementary measures? Education of the prescribing doctors. They must be aware of the risks that are in antibiotic overuse, correct use of existing guides, guidance of correct antibiotic administration, optimal dose, and switch. This last point is very important, switch from parenteral to oral therapy. The ability of the antibiotic stewardship team to implement proper antibiotic prescribing depends on the existing of a team capable of providing a recommendation based on microbiological results. And here I show you the ideal team: microbiologist, IT specialist, that means informatics, epidemiologist, infectious disease specialist. Also, the selective reporting of test results, and these facts allows the antimicrobial stewardship team to reduce the inappropriate use of broad-spectrum antibiotics and the associated cost. Here, I put you this as the final, the picture that is named Making Soap Washing Sheets, which is on the cover of the Emerging Infectious Diseases, which is the paper, the journal of the CDC Atlanta, appeared in November 2005. Why do I put this here? Because here you can see a very quiet image from the countryside. And this is a picture made by Grandma Moses, and it provides a window in the routines of a family farm where humans contact with animals, as you can see here, occur very regularly. Interactions that encourage the transmission of zoonotic disease. Back in 1870, the farmers washed the sheep after a few hot days in the river. Also, in the same river, they make hard and soft soap, wash the laundry, and house cleaning. In producing soap, they extracted from the sheep's wools the ingredients, and this practice dates from millennia. Evidence of this enduring method, combining animal fats with wood, be found in various ancient texts from civilization, including Roman, Greek, and Egyptians. In this picture, the message of this picture is a nostalgic scene, but soap washes aways not only dirt and grime, but also invisible viruses and bacteria that are on the skin. It does not kill microscopic pathogens, but the combination of soap and water helps remove them, thus helping us to reduce illness or infection. So, soap and water and common sense are the best disinfectants. And this is attributed to William Osler, and you will learn about him when you will study endocarditis, because there are specific manifestation Osler nodules. And this is one of the founder of the Johns Hopkins Hospital in Maryland, and referred as father of the modern medicine. On the other hand, although less frequently, people washing sheep can inhale Bacillus anthracis, a spore-forming drug-resistant bacteria with high fatality rates in humans and animals. The presence of domestic fowl, which appears to be ducks or geese, visible in this picture, is an array of other zoonotic risks. For example, psittacosis caused by Chlamydophila psittaci, atypical pneumonia, is transmitted to humans by inhaling aerosol dry droppings. Also, the fowl produce parasites. Also, birds can spread other diseases. And also, the presence of pathogenic avian virus is being monitored continuously. Chickens gather in front of a gray barn near a man bed next to a small white structure. And this is the cause of Salmonella infantis. Also, other animals, including dog and horses, near the main house on the left of this picture, could spread Campylobacter bacteria and Giardia duodenalis parasites. So, in this apparently naive style, caution is prudent, because the farm landscape in the painting also represents a shared ecosystem for humans and domestic animals. The conclusion is, within this space, no zoonotic pathogens are always present, which underscores the need for integrated. And the final message of this lecture is the One Health concept, surveillance and response. And if you remember, in a slide of previous slide, I showed you the presence of antibiotics in animal husbandry, in wastewaters, in in industry. So we are affiliated, all of us, to this concept, One Health concept. And the CDC Atlanta encourages us to be aware of this concept with the animals, home animals, farm animals. And finally, when you go in the hospital, it's very important that you do the hand washing properly. And in our hospital, at least in our hospital, we have very precise instructions on how we do have to wash our hands. And I respect you very much because I have been in your country and I know that at every point you have a sink and soap for washing hands. And I truly appreciate that. So you are one step forward because you have in your culture this manner. So I think here you have the references. And Miss Moderator, I think that I fulfilled my job. I used the time, and now the paper, the presentation is open for questions. Are you here, Miss Moderator?
Yes, Professor, I'm sorry, the signal is a bit poor in my
Okay.
place.
Okay, so as I told you, I finished my presentation, and if you consider necessary, I can take one or two questions.
Okay. All right.
I can't hear you, sorry.
I'm sorry. Okay. So thank you so much, Professor, for the amazing lecture. Today, now we are going to open discussion session for the student. So please raise your hand and state your name. And if it's possible, later you can put the question on the chat box also.
So first one is Albert Johannes, second
Yes.
one is Alzo, and the third one is Wilmark, okay?
Okay.
So, Albert.
Please, Albert.
Okay, let me introduce myself. My name is Albert, and I want to thank you for the opportunity. And I also want to thank Professor Roxana for giving us a very interesting lecture today. And I'm honestly interested in the drug development process and your current work. Recently, I read that antibiotics are more effective against gram-positive bacteria than gram-negative bacteria. For example, beta-lactams work by inhibiting bacterial cell wall synthesis. In fact, if we examine it physiologically, our immune systems contain complement systems that work more effectively against thin wall or gram-negative bacteria. And my question is, are there currently any antibiotics developed to target the cell membrane of gram-negative bacteria? Thank you.
I have to tell you that designing a new antibiotic is very difficult. Because the pharmaceutical companies have to test different products, and after that, the product must go through clinical trial, which may take years and years. In this time, gram-positive and gram-negative bacteria develop resistance mechanisms. So here is the balance between designing new antibiotic and resistance mechanism. And the ideal antibiotic means that is non-toxic, that is well adjusted by the patient and cost less money. But you can't find the ideal antibiotic active on the same range on gram-positive and on gram-negative. And as you mentioned, the bacterial cell wall is different. Okay, next question, please. Also.
Okay, thank you uh, for the time, Prof. Let me introduce myself. My name is Alonso coming here. I'm a medical student from Universitas Politécnica Indonesia. My student identification number it's at with 0 1 after starting the topic of pathogenesis of body fluid disorders. I would like to ask the following question. My question is, considering that edema, hydrocephalus, and the area all involve disturbation in fluid movement caused by change in hydrostatic pressure, osmotic pressure, membrane permeability, or drainage system. How does the body prioritize its compensatory mechanism to maintain fluid hemostasis? And why can these compensatory responses under central pathological condition paradoxically worsen the underlying disease instead of the restoring balance? Thank you, Prof.
You have a question that I invite you to write me by email because the answer is longer. So I prefer to answer it completely by email, and now it's too long to answer to you. But yes, I understood your point, and of course we must take in consideration different resistance mechanism when we give an antibiotic, and also the side effects you mentioned about hemostasis. Now, if I understood well. Of course, you must take in consideration the patient situation, and there are scores that we give to see if we can administer an antibiotic to a certain patient, because in severe condition patient, of course, we administer antibiotic, but it can be toxic, it can damage the patient, so the patient will die even if we give antibiotics. So we have to take in consideration the patient condition. Please write me by email and I will give you more comprehensive answer, but for the time being, I think you are happy with it. Please, Will Mark, the next question.
Okay, thank you, Professor, for the time. So first, let me introduce myself. My name is Wimar Petrusnai Purba. You can call me Petros. So I want to ask one question. So earlier, the professor explained that the antibiotic is more effective, changed to, from injection to oral. So why is that? Like, because the parenteral is like more effective, like it is the fastest to get absorbed in the body of the human than the oral therapy. So why is the injection is has to be changed to the oral? Thank you, Professor.
This concept, the switch therapy, belongs to international guides. These are generated after studies made on a big number of patients. First reason is that it's easier after you establish the serum level of antibiotic, is easier for the patient and for the doctor to switch on oral therapy. I'm sure that no one likes to make an injection. Parenteral therapy means injection. That means hospitalization, someone who must do the injection. So after you achieve the serum level, you can switch. This is what the guides are prescribing. So in every hospital, because first reason is easier for the patient, is easier to administer. This is the first reason that I have in mind. But of course, you don't switch the therapy if the patient requires or if he can't accommodate the oral therapy, because we are talking about neurological patients that have swelling problems. We are talking about ICU patients that are intubated and can't swallow. So for those patients, in any case, we don't switch. But for a normal patient that, let's say, stays in the hospital for 1 or 2 days, then is discharged and can continue at home by parenteral. In this way, you short the hospitalization, and that means big economy for the hospital budget. Okay? I don't see other hands raised. So,
Does anyone wants to ask Professor Philip? You can open another session. Okay. Maybe not yet in participant, Professor. If it's possible, may I ask one question, Professor?
yes, of course, please.
So in Indonesia, we face many farm products. A recent study in Indonesia shows that many farm products, such as poultry, like chicken, fish, has been given some antibiotics, Professor, just like what you present just now. My question is, as a consumer, how do we reduce the risk of getting the antibiotic from those products, because we know that we eat that every day, Professor.
That's a
Maybe that.
that's a very good and general question. And everywhere in the world where you go to the supermarket, yes, and you buy products from the supermarket, okay? Like also in Indonesia and everywhere. If you have relatives at home, if you have relatives in the countryside, you can eat products from them. And in this way, we call them bioproducts, like they are biological, ecological products that are not given antibiotics. But a large scale, you have two options. You can become vegetarian, don't eat any kind of meat, or you move in the countryside and start to raise chicken, have a small lake raising fish. This is the only solution. Besides the antibiotics that are given to those animals that are farm raised, there is another problems. They receive also hormones. That's why we are facing a very early sexualization both in boys and girls. Boys have mammalian gland because of the hormones given to the chicken they eat, okay? Girls have very early pubertal changes, so the situation is very complicated. What I can tell you is restrict the meat consumption, but meat comes also eggs and the derivative products like milk, eggs, cheese, everything, yes? And fish. I don't know, do you have fish husbandries in, I don't remember, because when I went in the restaurant, I haven't bought fish. I don't know how it works. Where do you buy fish? From the supermarket, no?
Yes, sometimes supermarkets, sometimes traditional markets. But recent study,
Okay,
yes, shows
traditional
that.
market. I think you can buy from, if you are sure if there is traditional market, because there are other problems. The products must be checked from the veterinarians. At least in our country, when you go in the market with the product, it has to be checked by the market administration, no?
Yes. All right. Thank you, Professor. I believe the participants have all asked the questions, and they
Probably
did.
there are some questions from the 6 participants that left, no? Because I
Oh,
see that, okay, from the participants that left, probably they had but okay, I'm very happy that the students got questions. I hope they are not inspired by the artificial intelligence, but even so, I'm very happy because the doctors that use artificial intelligence are okay. Those who don't use will be eliminated. So it's not a problem. So you recognize that you use artificial intelligence in your question? He haven't, okay. He missed my remark.
Alright, so adik-adik, apakah ada yang mau bertanya lagi? Cukup? Okay. So I think the student already understand the lecture, Professor. For the closing, I would like to say thank you on behalf of our faculty and all of the students. Thank you so much, Professor, for the insightful lectures. We hope that we can see you again in another lectures. And before we end the meeting, I would like to invite everyone to open the cam so we can have a picture together with Professor Phillips. Dedek, silakan dibuka kameranya. Mari kita foto bersama dulu dengan Dr. Philip. Okay, I'm going to start from screen one.
Yes.
We say cheers.
And also, maybe you can release me a certificate with your symbol of your university, certificate that I was the invited lecturer. Yes,
Yes, of course, it's
like
possible. I would,
last time.
of course, Professor. All
And
right.
I was very nicely impressed by the big number of students and their ability to ask questions and their interest showed in this presentation. And I appreciate that very much. And yes, this is the sign in Indonesia, no? I know, yes, I know.
Okay. Okay, we're going to start with the screen number two. Okay, screen number three. All right. Another screen. Okay, I think that's all. Okay. Thank you so much, Professor, for today's
Thank you. I'm waiting for the picture as well, okay?
all right. We'll be sending you.
Okay, thank you. Have a nice afternoon after that.
Okay. Have a nice day, Professor.
Thank you. Thank you. Bye-bye.
Bye-bye.
Thank
Thank you,
you.
Professor, for your time.
Thank
Bye
you,
bye.
Prof.
Bye bye. Bye bye.
Bye-bye.
Uite ce de inimioare aici.
Terima kasih banyak adik-adik untuk keaktifannya. Luar biasa banget.
Terima kasih juga, dokter.
Terima kasih, dokter.
Saya izin live ya adik-adik. Semoga kuliahnya, ilmunya terpakai untuk ke depannya ya. Oke deh.